So this class is titled Introduction to Computational Bioengineering. It's part of a 2-semester sequence. The first semester is going to be about relatively simple network models. The second semester gets into spatial modeling. It's going to be called Dynamics on Networks, and I'll explain a little bit about what that means in a bit. Historically, we've used a textbook by Herbert Sauro called Systems Biology, Introduction to Pathway Modeling. It's a very good textbook. It's heavy on chemistry. And so the detailed relevance of some of the examples may not be as great this semester. And I should say also that this class is streamed and recorded. So people should know that this is being recorded and is going to be distributed as part of the university. So the first class, we're going to see how far we get. It's a lot of new material, some old material, some new material. First, I'm going to explain a few words about why we decided to focus on virus modeling this semester. And then we'll talk about networks as a concept, a little bit on course logistics, and then we're going to get into doing a little bit of programming and exercises in class today. The structure of the class is typically, we've got 3 hours. The reason it's a little bit longer than the typical class is because we'll do in-class exercises. So it's a mixture of lab and lecture class. We'll typically take a break halfway through or 2/3 of the way through for 5 minutes. If I forget that, please should feel free to use the chat or voice to tell me, hey, you forgot to take a break, because sometimes we get distracted. And we're going to use just a very simple model today. Possibly we'll get a little bit further, but I think I want to get through at least the simple model today. I sent around a questionnaire, a little bit of a pre-assignment for people to get oriented. And then I also sent the first homework. They're going to be weekly homework assignments, which we can go over later. So the main reason we're going to focus on viruses in this class is the fact that the world has ended. And I think that viruses are of relevance probably to almost everybody. If you look at the death in the United States from COVID, it's an interesting curve. It's going to keep going up. It wouldn't be surprising if by the end of the year there were 300,000 or 400,000 deaths in the United States. I hope that's not the case, but it's certainly possible. We're going to be at 200,000 deaths by the end of the middle of September. So the factor of 2 either way is not going to be very difficult. COVID is, of course, a virus. It has many similarities with other respiratory viruses like flu or the cold. The cold is typically a coronavirus, so it's similar to COVID in that way. Flu has symptoms that are more like COVID. One of the things that's going to be very interesting this fall, which we may or may not get to, Josh here on our TA has been working on this, would be building some models of co-infection between SARS-CoV-2 and influenza. It's not exactly clear what's going to happen with co-infection, but there's some indications that it could cause very serious consequences. So that could be interesting to think about. So the general course topics, which are going to be unchanged year to year, are first, what is it that we can model using the concept of dynamics on networks? And what are dynamic network models and how do we build them? And how do they work? What are their basic properties? And then how can we analyze them mathematically, computationally, and to predict how they work? And all of those things are going to be quite general. The things that we do in the context of virus modeling would apply equally well if you were modeling chemical reactions. They would apply equally well if you're modeling economic systems. They apply equally well if you're modeling neurons. or if you're modeling gene regulatory networks or signaling networks inside of cells. The mathematics and the techniques, computational techniques, are identical in any of these examples. Typically, we do mostly signaling networks and gene regulatory networks in this class, but there really isn't that big a change to switch to viruses. I don't know, has anybody taken a class on this topic already? Or no? No. In general, people don't. What we'll find is that there's some math, there's some computing, but the level of sophistication of these components is not that high. And typically people don't come in with a background in this. And so we'll try to introduce all of the concepts as they come up. And if things are going too fast, please say so, and we can come up with additional material to cover details. So when we think about viruses, you can actually model viruses as networks in a whole variety of ways. The most obvious one, which is one that is used a lot, is in modeling epidemiology. How does the virus spread from person to person and cause infection? And when you see projections of how many people will be infected or die in a given country, when you see projections of how social distancing or mask wearing could change that, that's all done using the kind of modeling that we're going to do in class. Some of those models are a little more complex than the ones we'll do in class, but they're not fundamentally very different. We'll find that the modeling of how virus spreads from person to person is mathematically indistinguishable from the way you model the spread of virus from cell to cell once it gets into your body. When cells are infected by virus, they produce virus, and that virus spreads within your body and infects additional cells. And the pattern of spread is very similar to that occurs in infectious diseases at the epidemiological level. And already today in class, we're going to give an example which really could cover either one of those. Something that you have in the body that you don't have at the population level is the immune system and how the immune system responds to viral infection. And we'll do a little bit about that. The immune system is still not very well understood scientifically. And so one of the big challenges of understanding the lethality of SARS-CoV-2 and COVID-19 that it produces is that we really don't understand how the immune system responds to a virus very well. One component of viral modeling is the replication of virus within a cell. And one of the things that we'll look at is modeling both SARS-CoV-2 replication inside of cells and other virus types. Different viruses have rather different patterns of replication, depending on whether it's single-stranded, double-stranded, RNA viruses, DNA viruses, retroviruses. Viruses like influenza, the genome is RNA, but it's broken up into 8 separate pieces, and those replicate separately. With SARS, the replication is a single strand that's replicated as a block. Another thing that is quite interesting and relevant from a medical point of view and industrial point of view is what's called PBPK, or pharmacokinetic modeling. So if you take a pill, the availability of the molecule in the pill changes in your bloodstream in time. The pill has to be dissolved in your gut. It has to be absorbed through the lining of your gut. It's then distributed in your body, gets into tissues, and it's then cleared. Some drugs, in particular a drug like remdesivir, which is the one drug that's approved for treating COVID, remdesivir is given by injection, not orally, which is a problem. And it itself is not the active component. It's a metabolite of remdesivir that's the active component. And that changes the availability quite a bit. And so looking at distribution and clearance of a drug is something that one can do using this kind of modeling. When the virus gets into your body, typically it starts in your nose and throat and then moves down into your lungs. And there are models of this network kind that model the transmission of the virus between regions in your body. That kind of modeling is used a lot in all sorts of contexts, again, not only in viruses. One of the things we may be able to address, there's some very good papers by Alan Perlson, is the effectiveness of different drug treatment strategies. If you use remdesivir, how frequently should you give it? When should you give it? These are kinds of questions that can be addressed with this kind of modeling, and they're used in industry to design optimal treatment strategies. Something we may or may not get to, although there's one paper in the little stack already on this, is trying to study what happens after a lockdown. If you've locked down and gotten the viral load in the population to a low level, how do you unlock? How do you resume economic activities? And that particular modeling paper in question I've uploaded from Israel actually had a rather major effect because it seems to have been responsible for the government being elected that's currently ruling Israel. It's not often that modeling papers, mathematical modeling papers, affect governments, but that one did. And then we will inevitably, when we talk about biological context, talk about how cells synthesize molecules, because viral replication is about synthesis. How cells send and receive signals, because the immune response is about that. And how cells decide to differentiate and change their behaviors. If you want to understand what's happening with viruses inside of the tissue, you have to know those things. We'll find again that all of these examples and many more have the same mathematics and computing. And so the same technologies that we'll build mathematically and computationally to solve any one of these problems will apply to all of them. So the computing we do in this class is not that sophisticated. It winds up being enough. I don't believe that you should use technologies that are excessive. The simplest thing that works is the best. We're going to use a network modeling package called Tellurium, written by Herbert Sauro, you have the textbook, and Andy Samoji here at IU, which allows us to specify networks and their dynamics very, very simply. It's a text-based model specification. There are graphical interfaces available. And you can find it at tellurium.analogmachine.org. We're going to run Antimony and Tellurium in a web deployment using NanoHub. NanoHub is a server system available from Purdue. I hope that people have set up their accounts there already. We'll review the steps during class, but it's really critical that you get that. You could certainly download and run Tellurium on your local machine, but if you do that, it won't look quite the same, and other things will be slightly different. Particularly if you're using a Mac or a Linux box, you may have some issues. It works great on Windows locally, but not so wonderfully on other systems. And there's a big advantage then to using it on a server. The fitting, plotting, and data analysis will use Python, not terribly sophisticated Python, but basic Python. We will use NumPy and SciPy fitting tools. So some familiarity with that would be good. We'll bring them in as we need them. For a couple of specific things later in the class, we'll use one other package outside of the ones that are listed, which is a package called XPP-Auth, which was written by Bart Ehrmantraut at the University of Pittsburgh. And it's very good for certain specific kinds of mathematical analysis of what are called bifurcations, and for plotting phase planes. If you look in Herbert's textbook, every figure, except for 3 or 4 figures towards the end, were developed using Tellurium. All the figures in his textbook that were not using Tellurium used XPP-Auth. It's also extremely simple to use. So it won't be that much extra work to learn how to use it. And it happens that we talk to Bart every week, and he's teaching a course on COVID modeling at Pitt. So we could share some ideas with him as well. So in terms of programming skills, if you are not a very experienced programmer, don't be intimidated. There is certainly some programming, but it's not an enormous amount. I've uploaded onto both Canvas and the Google Drive that we'll be using. I prefer the Google Drive to Canvas. If you have trouble getting to the Google Drive, please let us know and we'll reshare it with you. A one-page Tellurium cheat sheet. All of the things you need in Tellurium could be put in actually half a page. You don't even need a full page for it. Python, there's about a 6-page introduction to Python that has the key ideas. Tellurium has a very, very good online documentation, which Josh has shared with you here. And I encourage you to read that and try the exercises, especially if you think that you might be interested in doing modeling of chemical reaction networks, for example. Herbert has excellent examples of that on the site. And almost any question that you need to have answered will be there. In the unlikely event that there are issues technically with Tellurium, as I mentioned, it was written by Andy Simoji here at IU and Herbert Sauro at the University of Washington. They're both on call so that if we have problems, they're usually overnight willing to help. How many people have used Python before? Has anybody not used Python before? Looks like everybody has. One person. Okay. So if you want a refresher in Python, I recommend strongly the Rosalind free tutorials. They're a lot of fun. I did them. They teach you how to do bioinformatics. And so it's a bioinformatics focused set of exercises. A lot of programming courses don't have exercises that are meaningful. They pick things sort of randomly. Rosalind focuses on doing classic bioinformatics calculations and teaches Python in a very nice way. So if you want a refresher on Python, I recommend that. It's got graded exercises, very nice. If people decide to do it, I always give a small amount of extra credit for that. But if people don't need it, that's fine. Occasionally we get people in this class who've never done any programming at all. Then I typically send them to Code Academy. Rosalind assumes that you have some basic programming, although not necessarily Python. So let's talk a little bit about what the subject of the course is, which is networks. And you can start out by saying, what's a network? We use the word a lot, and the world is full of networks of various kinds. And fundamentally, a network is there'll be a little, this next half hour, 45 minutes, maybe a little bit abstract, and then we'll come back to reality pretty quickly. And I'll crave your indulgence for the abstraction. There's always a tension between covering the general concept first and then the examples, in which case it seems very abstract, or covering the examples first, in which case the points may not be so clear, and then doing the general. So we'll do a little bit of both. We'll go both directions. So networks are an organizing concept. Many things in the world, including things that may not seem like networks to begin with, can be organized and analyzed as networks. And this class, again, will focus on biological networks, especially those related to viruses. But as I've said repeatedly, and I'll say many times, these techniques are general. They can apply to almost anything. Industrial systems, pipelines, economies, so on. And so if we think about networks, we can draw networks. Their networks are an abstraction, and the basic idea of a network is that there are nodes, usually here given as dots, and connections between those nodes, usually given as lines or links. And classic networks might include airline networks. This is a very old airline network for United Airlines from their frequent flyer magazine. I guess that may be so relevant right at the moment. The internet backbone connectivity network, again, a very old one. It would be a lot denser now than it is in this picture. Human protein interaction network, which is not very informative the way it's drawn. That's called a hairball for obvious reasons, showing all of the different proteins in a cell and how they interact with each other. And then the kind of thing that people like Cotty Berner here at Indiana University do, which is a citation network. So these are topics in philosophy, and the connections show how often they co-cite each other. Articles on one topic cite another topic. That's actually used quite a bit in analysis of scientific literature as well. Here's at the top right, we have a network showing world trade flows, also probably out of date given how much trade has decreased in the past few months. A human disease network. Now, this is not a network of infection, but rather a network showing relationships in terms of scientific literature between different diseases, showing the clustering of different kinds of diseases. And then a classic network of a different sort, which is a tree, which is the evolutionary tree here of more or less all life. Viruses not being counted as living organisms don't get into that particular tree, although there are evolutionary trees for viruses too. If you do an evolutionary tree for a virus, one of the things you discover quickly is that it's not a branching tree. It's not an open tree because there's so much recombination. Branches come back together. Neurons are a classic example of a network. The brain is a network, a complex one. And there are some very nice custom software packages like Genesis and Neuron for simulating the dynamics of neural networks. Of course, neural networks and all of the networks that are used in artificial intelligence and deep learning are a class of artificial neural network. Here's an RNA silencing network. So this is another molecular network for signaling inside of cells. And then at the top left, one that we'll address in some ways, although we won't address it in precisely this kind of situation, which is flu transmissions. And as you look at these networks, you could think about how they work. We're going to talk about dynamics more in a few minutes. But one of the things that you can ask about a network is the nodes, which are the circles usually, are those static or not? Do they move around? Is the linkage between components temporally ordered or not? So in flu transmission, somebody gives the virus to somebody else. And so every link between one individual and another has got a directional arrow which moves in time. On the other hand, if I look at a neural network, computational neural network, it's true that when I do computing, information flows temporally from the left to the right, but the connections are static and the nodes are directed but not intrinsically temporal. In RNA silencing, the coupling between two components is not directional, it's bidirectional, but it's combined. And there's no temporal order. In the case of a network which represents, for example, an airline route diagram, there isn't any temporal order in that network either. And so both the nodes and the links will differ in what they do and how they're represented depending on what it is you're representing. If we come to biology, there are four really classic kinds of network. Chemical reaction and metabolic networks, where molecular species combine, form new components. Those are the things that transform, create and destroy chemicals in cells, also energy. Signaling networks that transfer information within cells and between them. Regulatory networks that turn genes on and off and change cell types and cell type behaviors. And then at a higher scale, the transport of molecular species within your body, primarily through the bloodstream. And those are what are called physiologically based pharmacokinetic models. We'll definitely deal with the metabolic network when we do viral replication inside of cells. We'll do physiologically based pharmacokinetic models of drugs in the body. We'll do a little bit of regulatory network modeling. And Josh has been doing some nice work on signaling networks and the combination of regulatory networks in terms of viral resistance, interferon-based viral resistance, and we'll do that. So these are 4 classes of network. Again, structurally, they're identical. The time scales over which they operate, the names of the nodes, types of arrows and links they have, those will differ. But mathematically and conceptually, they're the same. Another kind of biological network that we're going to run into are population dynamics networks. In this case, we have, for example, a picture of a colonic crypt. At the base of the colonic crypt, you have stem cells. Those stem cells divide and reproduce. They also differentiate into early, here are labeled semi-differentiated cells that divide rapidly. Those cells in turn differentiate into terminally differentiated cells that don't divide, those eventually die. In the case of the lining of your gut, every cell in the lining of the gut, except for the stem cells, has a lifetime of about 2 weeks. So the cells actually physically move from the bottom of the crypt up along the wall of the crypt to the top, where they die and fall off. That takes about 2 weeks. This kind of model is called a population dynamic model. And epidemiological models, SCIR models, which we'll get to either today or tomorrow or next week, have exactly this structure. And it's one of the classic kinds of simple model that we'll address. And so again, the same model that you could use to represent the spread of virus in a population winds up being a model that could be used to represent the functioning of the renewal of cells in your gut. Another direction that we're probably not going to talk about so much this time are what are called adverse outcome pathway networks or outcome networks. And those are used very often for modeling toxicity. If you want to understand, is a chemical going to cause a problem? For example, you're developing a new drug, you want to know whether that drug is likely to be toxic. Then toxicity models are used for that. And those are quite interesting in their own right. And we may be able to do a little bit on that in class. Okay. So that's the first taste of what networks are. I give you a sense of how broad a topic they are. If we want to really understand how networks work, we have to know a little bit about a lot of things. We don't have to know a lot about any one thing, at least to begin with. But we need to know a little bit about a lot of things. We're going to have to know some biology, because we're going to use terms like DNA, RNA, proteins, viruses, cells. We have to know a little bit of biochemistry, because we're going to be talking about chemical reactions. We need to know a little bit about bioengineering, which is really the theme of the class. We're going to need to know a little bit about biophysics and how rate laws work. Definitely some chemistry, whether we do chemical reactions, some chemical engineering, tiny amount of computer science, especially when we do things like fitting. Data science, because we're going to be pulling in experimental data and analyzing it. Dynamical systems, which is really probably the single key idea, and which is something that many people don't learn, unfortunately, as a branch of mathematics. I know at least one person said they were taking, I think, maybe Randy Beer's course in dynamical systems, which is an excellent course in that area. I used to teach dynamical systems quite often. A little bit of mathematics. We're not going to go beyond first order ordinary differential equations, but you do need to know what derivative looks like in time. And we'll do a little bit of linear algebra in terms of analyzing stability. And one of the reasons I have that questionnaire about background is because depending on people's background, we do more or less mathematical analysis of these problems. And then some statistical background is useful, not anything very sophisticated, although we may try to do a little bit of likelihood analysis this semester, which is actually new for me too, and is pretty important. And so, unless you've taken a course in a specific area, or particularly dynamical systems, biological dynamical systems, I would expect that everybody in the room has at least one of those topics that they're not completely familiar with. And so one of the things that I hope we could do is have people team up to some extent and work together, because often we'll find, for example, some years we've had experimental wet lab biologists take the class who didn't have much computing and mathematics. And on the other side, we've had people who were very good at computing but had never had a biology course. And so people can team up and work together to learn from each other. I hope that this class will be like that. And because it's a different group of people every year, and because the subject area is a little bit different every year, if there's some area that's unfamiliar to you, please feel free to say, hey, slow down, cover that in more detail, review it. At least ask for literature background, which we can give. And also don't be ashamed of or shy about saying you don't know something or that I'm going too fast. Something's unfamiliar or unclear. If it's unfamiliar or unclear to you, it's almost certainly unfamiliar and unclear to other people in the room. Maybe it's worth, given that we've talked about mathematics, to say why this course is in engineering at all. Really, you could say that science is about discovering models. That is, models are principles of operation of things that could predict how those things behave under particular conditions. Engineering is really about taking that scientific understanding, those models, and applying them to either build new things or control things that exist. And so engineering is really about construction and control, and science is really about prediction. You need the science to do the engineering, but the engineering takes things a lot further. And I hope that at least by the end of the class, we'll have some examples where we can do some predictive control of the systems we're looking at. If we think about what we want to do with modeling in biology, first thing we want to be able to do, of course, is explain the mechanisms that lead to particular phenomena of interest. We'd like to be able to predict things that we haven't seen before. That is, not only post-dict things, but predict things. We'll see whether we get there. This class we may or may not. We'd like to be able to see if there are patterns of behavior that are shared from one place to another. That's really what the process of science is about. It's looking for shared patterns of behavior. And we'll call it what we call generic mechanisms. Biology is full of things. Engineering, human engineered systems, you build things like a screw, and every number 18 screw should be functionally identical to every other number 18 screw. Biology will run into concepts like cells and cell types. We'll have a concept of, say, a neuron. Every neuron is somewhat similar to every other neuron, but every neuron is different from every other neuron as well. And so the idea of replaceability of components is a very weak one in biology. It's much stronger in human engineered systems. So generic mechanisms are actually a rather complicated idea in biology. We'd like to be able to design experiments, guide experiments, interpret experiments. One of the things, especially in biomedicine, that's critical is that, especially if you're working on what's called personalized medicine, you can never do an experiment in personalized medicine. The core of science is what are called control experiments. You compare the result if you treat somebody with the result if you don't treat somebody. But since every individual patient responds differently to treatment, you can't treat and not treat the same individual. And so the core idea of science, which is control experiments, is not possible in medicine. The core idea of replication of experiments, that you repeat an experiment and see whether you get the same result, is also not possible. The idea that you give different treatments and compare the results in medicine is not possible. And so computation, if you could build a computational surrogate, what are called a virtual twin or digital twin of an individual, you can then do as many experiments as you want on that virtual twin. That allows you to make medicine scientific in a way that's not possible today. It's actually pretty shocking how unscientific medicine is, biomedicine in general. And that allows you to do things like try to optimize therapies. And in the case of COVID, for better or worse, COVID modeling has become very tied up with policy, political decisions, economic decisions, regulatory decisions. And so we're probably not going to go into the weeds on policy. But for example, the work we've done on toxicology, predicting the toxicity of drugs, is something that has policy implications. Population models of epidemics definitely have policy implications. One of the things we're going to find when we start digging into epidemiological models is how primitive the models are on which policy decisions are made. It's a little bit shocking. Okay. So again, coming back to this idea of why are we in engineering here, if we're going to be able to do successful engineering, we have to be able to predict the effects of what we do. If we're going to say we're going to change something to have an outcome, we have to be able to predict what the effect of that change is. And that's really not easy to do when we're dealing with living organisms. There's a famous saying that under carefully controlled experimental conditions, an experimental animal will do whatever the hell it wants to. And that is an issue in biology. Replication and repeatability are not as simple in biology as they are in other sciences. And so we might be able to, for example, want to predict if we apply different social distancing rules, what fraction of the population will die of COVID. And we'll come back to that in just a minute, and we'll see some predictions and how different they are. It shows how much uncertainty there is. Another place that you really would like to be able to do prediction is in therapy. We've got remdesivir as a therapy. Antibody treatments have been in the news in the past day or two. Anti-inflammatory medications like dexamethasone have been in the news in the past day or two. It winds up that while in principle we understand what they do, in fact, how to apply them to save people and not kill them is actually rather difficult. And it may vary from individual to individual. And so we'd like to be able to actually design and predict what's going to happen before we treat somebody rather than just try our luck and see what happens. Okay, so that's a high order view of networks, and we'll come back in a bit to a little bit more detail. We'll review that material again. Any questions about that so far? You're free to unmute if you want to ask a question or use the chat either way. But I hope that gives you a little bit of a sense of where we are with this. Okay. So course logistics. I'm teaching Josh, who's helped out for years, officially or unofficially, and who took this course many years ago, is TA for it officially this time. We're meeting for 16 weeks, as far as I understand, not 13, unless there's a huge groundswell of enthusiasm for cutting the course short, 16 weeks. I don't know what office hours really mean in the context of COVID, but if people want to talk to me by Zoom, I'm available as much as I can be. As I mentioned, the main textbook we've been using in the past is this Herbert Sauer Systems Biology, which is excellent. I encourage you to buy a copy. You can download a PDF for not very much money. You can also buy a printed copy. I strongly recommend, because I think this year we're going to be doing a little bit more mathematics, getting a hold of Nicholas Britton, Essential Mathematical Biology. When I used to teach dynamical systems to undergraduates, it's the textbook that I used. It's short, it's cheap. You may even be able to get a free copy if you look around. And it has the basics in a very compact and accessible form. In the past, we've used Eberhard Voigt a little bit. First course is systems biology. I don't know how relevant that's going to be this semester. Herbert has some other good textbooks that cover details. There's a textbook which we just ran into the other day, which looks very good, and which seems to be available for free online, which is Brouwer, Mathematical Models in Epidemiology. And I encourage people to take a look at that. And then we're going to have a significant number of scientific articles that we're going to use as references. There's going to be a piece of the textbook available temporarily, but with Herbert's textbook, I really need to insist that you buy a copy. So it's there until you can get your own copy. It's not fair to Herbert to preempt his $20. It's not a $250 textbook, it's a $20 textbook. So we have to encourage Herbert. Herbert's worked closely with us over the years in the sense that every year we send Herbert a pile of corrections based on our experience teaching from his book, and each year he incorporates them. So every year it's a different book, much improved this time. And so I do encourage you to use that. But I will make at least pieces of it available until you can find it. You can get it immediately as a download from that website that's listed there. Course logistics, grading, they're going to be weekly homeworks. They're going to be probably half of the course grade. There'll be a mini project. That'll be, in a sense, the most important thing in the class, which will be your either developing a model of your own or taking an existing model, implementing it, and extending it. People are welcome to work on that project in teams. 2 people together is fine. I'm going to ask you very specifically to take that model and deploy it on NanoHub as a NanoHub application. And so part of your grade will be on getting it deployed on NanoHub. And I encourage you to get involved with figuring out the logistics of deploying it on NanoHub early, because it's not so trivial. And we have Giuliano who will come in and help us on that. He's the local expert on that. The plan is that there'll be a midterm exam, a small midterm exam. Depending on how the class works, that may or may not be practical, so we'll have to see. There'll be an oral presentation of your project. People will be graded a little bit on class participation. If you do decide you need to do some background computing bone up, you'll get a little bit of extra credit for that. No final exam. I don't believe in exams very much, especially for a course like this. But I do really want people to keep on top of the homeworks. If you get behind, it's hard to catch up. And on the projects, I'm also going to have to be pretty strict about keeping people up to on time. These are projects that you can't do in the last week. You have to get started on them right away. So next week I will hand out the project assignments and ask people to work on those starting from the very beginning. It's really, these projects are they're not technically very demanding, but they do take time, and figuring them out takes time. They're not things you can do at the last minute. I strongly recommend, given that you need to know a lot of different things that are not very high level, starting at Wikipedia is always a great thing. Wikipedia tends to be well written in the areas we're going to be working in. We've got good articles on network theory, network science, dynamic network analysis, all good articles. If you want to learn about stability analysis and parameter identification, Wikipedia has very good articles. Depending on your biological background, reading about viruses and how viruses replicate on Wikipedia is not a bad idea. Reading about how the immune system works is not a bad idea. Reading about how epidemiology models work is not a bad idea. At IU, actually, some of those people have left now, but there was, for a while, probably the world's strongest group in epidemiology modeling was here at IU. Alessandro Vespignani, who's now at Northeastern, was here. Alessandro Flamini, who started out as his assistant, is still here. Over the years, they've had a lot of people graduate doing epidemiology models, rather more elaborate ones than the ones we'll do in this class. And I remember sitting a couple of years ago, well, now several years ago, on a PhD committee for somebody who did his PhD with Alessandro, and then went to work for Bill Gates. His PhD was on influenza epidemics modeling, and Bill Gates hired him personally. I don't know if he's still working for Gates, But when he came back to do his defense, his thesis defense, he had to get permission from Gates personally to have 2 days off to come back and do his defense. So this kind of thing gets, this kind of modeling actually gets used. And so the things you'll learn here are good entrees into that world. We're going to have a folder on virus modeling, and one of your assignments for the first homework will be to do a little bit of Googling, find a virus model or virus-related model, and read enough of it to be able to explain what it is and then upload it to our shared repository. So I'd like to come back to a few general ideas that we're going to run into. Some of this, again, will perhaps seem rather abstract, but there are ideas that we'll come back to. The first one is what a model is. And a model is something that predicts a set of measurements that you can do on something that you could measure, at least in principle, in experiment. And it depends on another set of measurements that you could at least in principle do. And a model has 2 components to it. There's something called a model structure, which is the architecture of the model, and the model parameters and variables. And so a model is something that takes a set of input parameters and model structures and combines them to make a set of predictions. And we'll see that very clearly because when you write things in antimony language, you have a block of text that defines a model structure, and then you have a block of text that defines model parameters, and a block of text that defines variables. And so that breaking down of a model into 2 components, which is a model structure and a set of parameters, is something that may seem rather abstract, but it's going to be quite important. A lot of the problems that people run into when they try to do mathematical and computational biology, because they don't make that analysis into pieces that way clearly. And so I'm telling you about this now because we're going to come back to it again and again. Models are always abstractions. They're very extreme simplifications of reality. Exactly how extreme we're going to see when we start doing epidemiology models. It's almost impossible to believe how oversimplified they are. And yet they do make potentially useful predictions. And one of the things that we're going to run into is that we want models to be detailed enough to predict the things we care about, but not too detailed. And there are going to be some mathematical frameworks that maybe help you make those decisions. But in fact, there's no universal way of doing that. But when you're designing models, the hardest part is to decide what to leave out. Not so much what to put in, but what to leave out. And we'll see that frequently once we go through the class. Now I'm going to talk about networks again. We've already seen some example networks. In past years, I would have had a little in-class exercise about networks, but I think we're going to pass over that one this time because I want to get to some computing today. So a network is a graph, and it's a model of some system. And that sounds abstract. It is. But the key thing about a network is that there are nodes, subunits, here little circles, and a set of connections between the nodes, links. And so that concept of topological arrangement of nodes and links is a network. What the nodes and links represent will depend on what you're trying to model. And you can have very, very simple networks. We're going to deal with some very, very simple networks today, networks with only 2 or 3 nodes. And you can also have networks that have millions or billions of nodes. You can have networks in which the number of links is many times greater than the number of nodes. You can have networks in which the number of links and nodes is almost equal. You can have what are called directed graphs, undirected graphs, hierarchical ones, trees, random networks. And there's a lot of mathematics that's been done about the topology of networks. That is, what's the shape of this mass of connections? And people like Alessandro Flamini and some of the other people, especially in informatics, spend a lot of time doing analysis of network topology. And that's specifically not what we're going to do here. We'll talk a little bit about it, but that's not going to be the focus of this class. So if I think about nodes and links, we can have, if we have a picture here of nodes around the edge of the circle, and here we have links, each one of these links connects two and only two nodes. And in many cases, that's how networks are built. For example, if I'm doing a classic neural network, I have one link between two nodes always. If I have chemical reactions, on the other hand, I could have three components coming together to make two components, in which case my link would have three inputs and two outputs. And so you can have rather complex links with multiple inputs and outputs. And so again, the particular pattern that you're going to have will depend on what it is you're trying to model. Network models are, again, highly oversimplified. When we are building models, what we're doing when we build a model is we're looking at some kind of real world situation and we're trying to infer an abstract model behind it. And that's really doing reverse engineering. When we're doing engineering, we typically know what the model is, the network is, and then we try to build it. An integrated circuit is a network. It's a pattern of transistors and interconnections that does some function, like build a memory chip, build a microprocessor. And so in that case, you actually have a computational model of that network. It's actually been simulated in great detail using a program called SPICE, which goes back 40 years now. New versions of SPICE are much more sophisticated than the old ones, but it's still SPICE. And then it's turned into a physical realization. So in science, there starts out with a real world physical object that we're trying to infer what the network is. In engineering, typically, we have the network and we want to understand how to build it. Networks can represent real world things, like the transistors in your computer. They can also represent things that are very abstract, like the network of citations between journal articles. Sometimes those objects have spatial relationships. The transistors in your computer have physical location and therefore a physical spatial relationship. Other times they don't. Chemicals in a chemical reaction network don't have physical relationship to each other. One of the things that you have to remember when you're dealing with a network model is that it's not intrinsically a spatial model. You lose spatial information when you build a network model. And that may or may not be important. One of the things Josh is working on a lot is the difference between a model of viral replication and immune response with spatial components and without. We'll find in this class where we won't deal with spatial relationships, we can get amazingly far ignoring space. On the other hand, we exist in space, and there are certain situations in which spatial relationships are critical. And so it's important to know when spatial relationships are going to change the results and when they won't. It's possible sometimes to build surrogates for spatial relationships in networks. And we may talk about that a little bit later on. In these pharmacokinetic models, there often are surrogates for spatial relationships. And when we model immune response, where we have cells that have to go back and forth to the lymph node, we'll have surrogates for spatial relationships as well. But in general, networks are topological models, not spatial models. Okay. So the nodes in a network are usually things. They're the things being modeled: cities, airplanes, chemical species, people being infected by virus. Nodes always have states. They have properties associated with them. Those properties could just be, say, the name of the city or the number of airplanes sitting on the runway of that city. Or they could be something very complicated. If the network consists of the cells in your body, the state of the cell might consist of a list of every chemical in the cell and the spatial relationships of every chemical in the cell. So there's huge variability in terms of the level of sophistication, level of detail of a given network model. However, the concept of nodes and states is still critical. In general, we don't have structure inside of nodes. A node is thought of as an indivisible object, although it's possible to build hierarchical models where you have submodels inside of nodes. If we're doing chemistry, it's called the stirred reactor model, which says there isn't any structure inside of the things that we're looking at. Links define relationships between nodes. And in the case of the dynamic network models that we're going to deal with, they define how things affect each other. They define interactions. And an interaction changes some aspect of node state. If a link represents the flight path of airplanes between Chicago and Kansas City, then that link has the property of taking airplanes from Chicago and moving them to Kansas City. So it changes the number of airplanes in Chicago and the number of airplanes in Kansas City in time. Typically, links are indicated in a dynamic network in the form of arrows, which indicate the direction of flow of information or material. Sometimes they're bidirectional, sometimes they're unidirectional. Sometimes they're mass related, sometimes they're only information related. But in a dynamic model, there always is some kind of information flow between the components. In a purely topological network model, like a citation model, there isn't. At least it doesn't have to be. Nodes, again, are usually indicated, and links are indicated generally by arrows. There are certain conventions for how arrows work. Unfortunately, there are lots of competing conventions, sometimes using opposite assumptions about what they mean. I'll try to be relatively consistent about arrow notation. And the arrow notation we'll use is based on the standard one in biology. But in other fields, the arrow notation is somewhat different. And again, arrows often have a property of transforming or transporting node properties. So moving airplanes from one place to another, moving people from one place to another. A link that represents my coughing on you might transform you from being uninfected to being infected. A link might represent that you died, so your state went from being alive to being dead. Those would all be transformations, changes of state, one sort or another. Coming back, you'll find lots of different representations of links and nodes. Some fields are strange. In economics, the nodes often represent interactions and the links represent states. I don't know why economists do it backwards, but they do. Almost every other field does it the other way, but if you read economic literature and you see network models in economics, they often have the opposite convention of nodes and links. Are there any economists in the room? No? Okay, so that's not going to be a problem. So networks, again, we're going to have different numbers and categories of nodes. You could have a situation in which nodes are all of the same class. All of the nodes are people. All of the nodes are cells. All of the nodes are virus particles. Or you could have a situation in which different nodes represent different types of object. One class of nodes might represent people, another might represent cars. A third one might represent horses. A fourth one might represent cities. So you can have nodes and models in which all the nodes are homogeneous in some way or all different. And again, what states those nodes have can differ depending on what you're trying to do. Whether the nodes have substructure, for example. The number and categories of links are also going to be different. In biology models, typically you have at least two classes of nodes and at least two classes of links. The links represent chemical transformation. Molecule A turns into molecule B. And regulation. Molecule A changes the rate at which something happens, but isn't used up in the process. Those are two fundamentally different ideas that have to be thought about and represented. In fact, in biology, there are a lot of different interactions, and there's a model, a standard for representing them called biotapestry that has dozens and dozens and dozens of different representations of nodes and links. Typically, we'll only use a couple of those here, but we'll try to follow those standards. And there are some cases where actually arrows point not to link to nodes, but to arrows. Regulatory arrows would be an example of that. So if you're going to die as a result of being infected by COVID, and I treat you with a drug, and the result of the drug is to reduce the death rate, I could represent that drug as an inhibitory arrow on the arrow that connects you being alive to you being dead. That would be an example of that. And we'll see that quite a bit. So we've talked about networks. Now let's talk a little bit about dynamics. Just generically, what are network dynamics? Networks have an architecture, topology, which we've talked about. And as I mentioned, there are a lot of people who study the fixed topology, the fixed architecture of networks. In our case, we're going to always be caring about how things change, how networks change in time, and we want to be able to control those changes. And there are two fundamental ways in which a network can change. The network can change architecture, that is, the pattern of connections, the number of nodes, the links between nodes can move around. That would be the middle one here, called dynamics of networks. And infection modeled as interactions between individuals is a dynamics of networks problem, because at every instant you're interacting with different people, and therefore the pattern of connections that you have is changing moment to moment. And what I care about is who I'm connected to at any given time and how that pattern changes in time. That would be a classic dynamics of network problem. Dynamics on networks says that there is a fixed pattern of connection architecture, network architecture, and what's changing are the states on those network nodes. If I say that my network consists of living and dead people, and there's a transition between living and dead people, what's going to change is the number of people occupying the living person node and the number of people occupying the dead person node. But the identity of the living person node and the dead person node doesn't change. And the fact that there's a link that connects living people to dead people doesn't change. And so you have a situation which is dynamics on networks, which is what we're going to focus on in this class, where the network itself is fixed architecture, but the node state evolves, and dynamics of network, where the network architecture changes in time. The big difference is that network node state evolution, dynamics on networks, is continuous in time. You can't make dynamics of networks continuous in time. Although the neural network people try, because they build networks where everything is intrinsically connected to everything else, and you set a lot of those connections to zero. So then you can change the connection strengths dynamically. And if you take Randy Beer's class, which is excellent, one of the things that he's studied in great detail is why, for example, in neural networks which have all-to-all connectivity, you never force the values back to zero. You never disconnect nodes. And then you have potentially situations, again, like the viral infection model of person-to-person transmission. Traffic models in the city would also be like this, where both the structure and the states on the nodes change in time. We'll do a little bit about that, but primarily we're going to be dealing with dynamics on networks. It's the simplest case. Computationally, it's also more tractable, but we certainly can address the elements. So we're going to focus on dynamics on networks. And so now we have to think a little bit more about that. So we've talked about node state in the abstract. I've given you some examples, but suppose the nodes represent chemical species in a cell. The state would be how much of each species we have. The nodes might represent the bank accounts in a bank. The state would represent how much money is in each bank account. The nodes might represent people in the population. The state would be, do you have flu or not? And these days we might ask, how many days has it been since you got the flu? And how sick are you? What's your fever? We gave the example of airplane network, number of airplanes at each location, at each airport. How many tourists you have in the city. That's a big issue these days, right, with COVID. Population of the city. But if it was a neural model, it might be the firing rate of a neuron as a function of time. If it's a cellular model, it might be the differentiation state of each cell in the system during time. And a node can have many, many state variables. It doesn't have to be just one. Again, in a dynamic network model, a link represents an interaction, and an interaction tells you how the states change as a function of time. In particular, how the states change as a function of the state of the objects that are being connected. Okay. And so dynamic models that we might address would be things like models of economic production and trade, neural activity we've talked about, epidemics we've talked about. There are lots of network models of ecosystems. Pollutant flows in the city, blood transport we've talked about a little bit, gene expression, which we could talk about more, chemical reactions, signaling between cells as well as within cells. One of the things that's worth noting about almost all of these here is that in reality they exist in space, and so a network model loses the spatial relationships, and that may or may not be important. And one of the interesting things to understand between this semester and next semester, if anybody sticks around for next semester, would be taking these models that we've developed in a non-spatial context, putting them into spatial context and asking how does it change the result. So again, to summarize this, any network model, dynamic network model, has nodes. The nodes have states, variables. There are links between nodes that define how those state variables change. And those links are described by rate laws. Those are laws that describe how the variables change. And that fundamental mathematical description is going to be the same for every one of these models. And that's why fundamentally the mathematics that we're going to do in the context of viruses could apply to neural networks, it could apply to economic systems and everything else. But it'll be different are the rate laws. The form of the rate laws will be different in these different models. But fundamentally, everything is the same. Okay, so it's actually, we got through that a little bit faster than I expected. Shall we take a break now for 5 minutes? Is that is it a good time to take a break? Or is it too early? Because now we're going to jump in and start actually doing a little bit of modeling and coding. So this is a good question. That the abstract stuff we're done with now. That's going to be most of the abstract stuff for the class. Do people want to take a 5-minute break, or are you okay? I'm not
Either
hearing.
one sounds good to me.
Sorry?
A break or not, it doesn't really matter to me.
Doesn't matter? Okay. I'm okay to keep going if you are. It's more, I guess if you're at home, maybe taking a break is less essential, but I know when we have 3 hours of class in a stuffy room, it seems pretty important to take a break. The one thing that I can't do is that traditionally I bring either cookies or candy or some snack so that we have snacks at the break. I figure that late afternoon, a long session, people need some sugar, and I can't give you the caffeine or candy remotely. So I apologize for that because I always bring snacks for class. It's one of the things that I always do. So that's the something I'm afraid I can't do remotely. Okay, so what I'd like to do now is introduce Tellurium, a modeling framework that we're going to use. As I mentioned, I hope it'll seem simple. It has its frustrations and annoyances like all modeling frameworks, but I hope it's not too bad to use. And we're going to build an exceedingly simple model of viral infection. And then we're going to start with that and build up. And we're going to use it as a platform to understand some mathematics and computing. As I say, I know that not everybody in this class is interested in viruses, and I want to keep emphasizing that the things you're learning here will generalize. So let's think about epidemiology, dynamics of viral infection. And one of the things that's interesting about epidemics is that if we look at the pattern of infection, for example, the rate of new infections versus time, we see a similar pattern, which is that you see a rapid increase of the number of people infected. It reaches a peak, and then there's a slower decay, and eventually it goes away. Now, in the case of COVID, COVID is what's called a primary infection. If you have never seen a virus before, your body's immune system takes a while, typically 7 to 10 days, before it really is able to respond using what's called the adaptive immune system. Once you've been exposed to a virus, if you're exposed to the same virus a second time, sometimes, if we're lucky, this is going to, the world ending or not ending will depend on this, in this case, your immune system remembers that it's been exposed to that virus. And in that case, you have antibodies and CD8 plus T cells that respond to the virus very rapidly, which means that the second time you're exposed to the virus, you either don't get sick at all or your illness is much weaker. One of the problems with immune memory is that it doesn't last forever. We really, really don't know what makes the difference. In other words, we don't know why exposure to some viruses leads to lifetime immunity, in which case also vaccination works extremely effectively. Smallpox was so effective that the smallpox vaccination got rid of the virus. Some diseases like measles, and measles is the opposite. Measles reset the immune system so that any immune memory you had before you got it was erased. Mumps was a disease that you got once, never could get again. But there are also diseases, for example, like influenza, which you can get repeatedly. Unfortunately, coronaviruses, like the cold-related coronaviruses, don't seem to cause a lot of immunity. You can get a cold, and then 4 months later, you can get the same cold. And so in terms of whether you'll ever be able to have a vaccine, that's a big deal. There are two issues with viruses, one of which is it the same virus? In the case of influenza, if you have a flu shot, the flu shot is specific to a particular strain of the virus. And if you're then exposed to a different virus, that flu shot either provides only partial protection, or there actually is can be situations in which if you're vaccinated for the wrong strain, it can actually make you more susceptible to other strains. Effectively, what happens there is that your immune system has a finite bandwidth. And if you tell your immune system to put your effort into protecting against virus A, and then you're exposed to virus B, it can actually downregulate your protection against virus B. And then there's some weird diseases like dengue, which is infamous because the first time you get it, it's not bad. The second time you get it, your immune system kicks into overdrive and can kill you. So the idea of building a vaccine for COVID is not crazy. It's likely that they'll be able to get something out of it, but it's quite possible that there will never be a fully effective vaccine for COVID. So there's a difference between primary exposure and secondary exposure. With flu, almost everybody in the world has had the flu at least once, so people aren't typically immunologically naive. However, the pattern of infection for flu winds up being rather similar to that for COVID. And so this pattern of an epidemic where you get rapid exponential growth, a peak, and then slower exponential decay is pretty typical. Sometimes it goes to zero or nearly zero. Sometimes it goes to a baseline. In some cases, you get periodic oscillations. In the case of flu, there are two peaks, one in the fall and one in the spring. And those happen annually. And flu in particular is a very complicated spatial pattern of spread through the world. It's quite interesting. Obviously, with the virus has somebody who's sick. That person transmits it to somebody else. You've probably seen a discussion of the R number, which is the typical number of persons that a person infected infects. So if each person who is infected infects two more people, then the number of patients who are infected doubles and doubles and doubles. That's how you get exponential growth. Clearly, at some point, either everybody in the world is infected, in which case you run out of people to infect, or something else happens. And so that exponential growth can't go on forever. It has to saturate. What happens after saturation is a little bit more interesting. Why it goes down is a little more complicated. But that's the typical response. And so here, worldometer, you could go to the Johns Hopkins site or the worldometer site. I like a worldometer. Is a pattern of infections. This is Italy. I picked Italy because Italy has been able to tamp out the virus pretty effectively. You see starting around February 24th, beginning of infection, peak of 6,000 cases a day around March 22nd. So that took about a month to get to the peak. And then a slower decay, March 22nd up to, say, May 24th to come back down. March, April, May, so 2 months. So the decay was about half the speed of the turn on. And then things stayed pretty low until they unlocked. And now the amount of virus infections is ticking up again. If I look at the United Kingdom, which didn't lock down very severely, I see that the same pattern occurs, but it's spread out over a longer time. It never got down as low, and it stayed a little bit higher. If I look at total deaths in this case are basically about 1% of the number of infections. This is essentially the integral of this curve here. So the slope of the total death curve is approximately the number of new infections lagged by a couple of weeks. If I look at the United States, the growth curve of the United States is slower. It's got a pretty flat top like the UK, not as peaked as Italy. It doesn't come down very heavily. It's gone back up in the past month or so. And here on the right-hand side of this, you can see projections of what the likely caseload is. This red curve here is what's predicted to happen if people keep going as they are, don't wear masks and don't social distance. This middle curve is what happens if people social distance a little bit more. And this lower curve is what happens if people actually got serious about trying to get rid of the virus. Given what's happening here in places like Bloomington, with people not wearing masks and not being very careful, we're likely to be more in the top range than the bottom. But if you look at here, the number of infections here is 100,000 on the bottom one and is 1 million at the top. So there's a factor of 10 difference in the prediction of number of infections per day. It's a huge uncertainty. And one of the problems with a disease like COVID is that scientists are used to uncertainty, but they want to be able to quantify the uncertainty. You want to be able to say, I know this value plus minus 20%, 30%, 50%. But to not know how many people are going to get infected on a daily basis by a factor of 10 means effectively we know nothing about what's going to happen. And that level of uncertainty is something that scientists and engineers are very uncomfortable with. And we're going to face that a lot when we try to do epidemiology modeling. In particular, when people tried to do epidemiology modeling early, their predictions were all over the map. because there simply wasn't enough information to make a prediction at that point. And I would say that right at the moment, we still don't really know enough to predict what's going to happen. There are too many uncertainties. Okay, so that's virus as an epidemic. If we now jump inside your body, and this is going to be mainly about flu rather than about transmission of virus from individual to individual. Now I have individual cells, and the equivalent of the uninfected individual, the susceptible individual in the population, is a cell sitting in your nose or your throat or your lungs that could get infected. And they're called T cell, T here for target cells. They come in contact with the virus. There's some probability that those cells become infected. That's an I1 cell, an early infected cell. It winds up that just the way if you're infected with COVID, your body will go through a couple of stages. For a couple of days after you've been exposed, the virus will be replicating in your body, but you'll not be shedding it. You won't be infectious. Exactly how long that isn't clear. Longer is better, probably. Then you'll have a period of a couple of days when you're infectious, but you're not sick. You don't know you've been infected. And that's the main time that people spread the disease, because they're running around not knowing they're sick, and they're spreading the disease. And then you'll get sick, and if you really get sick, you'll stay home and you'll stop infecting people. In the case of the cell has what's called an eclipse phase, where the virus is replicating inside of it but isn't being spread. An I2 phase, a late infected phase, where it's releasing the virus. And eventually that cell will die. Every cell that's infected in your body with the virus will die, unlike people who can recover. This cell will release new virus, and that virus will spread within your body and cause new cells to become infected. The basic pattern of this is not very different from what's happening in the epidemic population. And if I look at the viral load, the amount of virus in the system versus time, it's not identical to the number of I1 cells versus time. That would be a better surrogate. I see some particular patterns. The amount of virus in your body will increase exponentially after you've been exposed. There's a delay. First there's a delay, and then it'll increase exponentially. It'll reach a peak, and in simple cases, it'll then decrease, and that decrease will be also exponential, but at a slower rate than the one that happened on the uphill side. There are some situations in which you get different shaped curves. Just the way I looked here, the shape of the Italian curve and the UK curve was somewhat different. The US curve is different from that. There can be different curve shapes that you see for different viruses. COVID has this monophasic decay, typically. Influenza has what's called the biphasic decay, like that. And there are other diseases that have different ones. In some cases, you never clear the virus completely. It sticks around, and that's called a chronic disease. Classic chronic disease would be HIV. Once you're infected, you never get rid of it. It's not a virus, it's bacterial, but tuberculosis is another disease you typically never clear. And there are a number of viruses that stay integrated, especially DNA viruses tend to integrate in your genome. Once you've had them, you've got them for life. They may not make you sick, but they're there forever. They can break out at any time. And so there are a variety of different patterns of things that can happen. But fundamentally, what I want to say is that the mathematics and the structure of these models is pretty similar. Okay. There are a lot of models of viral infection, which we'll dig into, just as there are a lot of epidemiological models. These are all models of viruses within your body. I could show a similar set of networks that would be describing infectious disease models at an epidemic level. All of them consist of a bunch of nodes and arrows. And if I take this one at the top on the right, which is a model of Erica Mohan and Bart Ehrmantraut, who are people we collaborate with. We're working with them on this particular model at the moment. what you'll see is that there are circles and triangles and square and diamonds and octagons and other shapes, and there are a variety of arrows. And each one of those triangles represents a different thing. Let's see if I can tell from this, just looking at it, what they are. Let's see, V stands for virus. This diamond represents virus circulating in your body. It's going to infect cells. Let's see where the infection is happening. These circles, H, I, and D, represent cells, and the triangles represent chemical species. Arrows represent movement of material. These T-shaped arrows represent inhibition, and they don't seem to have distinguished activation from mass transport in their diagram. So this is a model that's moderately complicated. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, it's only 20 nodes. And probably 30, 40 arrows. This is a simpler model. This is a model of Alan Perelson, one of the great modelers of viruses. In this case, we have susceptible cells, which can become infected, and there are various models of what the infection is doing, and virus here. And so here I only have 1, 2, 3, 4. There's no box here for virus, although it should have a node of its own. 5 nodes, and I have how many arrows? 1, 2, 3, 4, 5, 6, 7, 8, 9, 14 arrows. Each one of these models then is a network of varying complexity, and we can come back and look at the details. Sometimes the models will be so simple they don't really look like networks. If I only have two boxes and 1 arrow, that doesn't look like much of a network, but it's the beginning of a network. Okay, we're going to talk in class, we probably won't get back to it so much today, about what are called SIR models. These are the simplest epidemiological models. Susceptible, infected, and R has a sort of a funny usage. R can stand for recovered or resistant. It also includes dead. So in an SIR model, the assumption, and this could apply both to cells in a tissue or individuals in a population, is that you have a group of cells that are uninfected, which are S. And inconveniently, those are often called T rather than S as a variable, which are susceptible. There are things that can be infected, cells or people. There are individuals that are infected, I, infected, and those infected individuals can infect others. And then they're going to be recovered or dead individuals. Sometimes it's called SI, susceptible, infected, and removed. And the key thing in this SIR models is that the recovered individuals can't be reinfected. So once they're recovered, they're out of the group of infectable population. And a virus that you can't get twice, like smallpox would be one, or mumps would be an example of that. So susceptible individuals become infected when they're exposed to the disease. Infected individuals eventually recover and maybe die, but in any case, they're taken out of circulation, and they're not able to be infected again. And so we have a teeny little network, which consists of 3 nodes, S, I, and R, and 2 arrows. S goes to I goes to R. You can't go from being susceptible to resistant immediately because it requires being infected to do that. You can't go backwards from infected to susceptible, at least in this model. And in this model, once you're resistant, you stay resistant. They're what are called SIRS models, where the resistance individuals lose resistance after a while. That would correspond to your immune system gradually fading away, your resistance fading away. and becoming susceptible again. This model is what's called mass conserving. It works like a chemical reaction because every individual in the population stays in the population. If I start out as a susceptible individual, I either stay a susceptible individual forever or I become infected and then become resistant. I don't have any source of new people. There's no arrow coming in from the outside. And I don't have anybody going out of the system. I don't have any arrows pointing out to nothing either. And so this arrow diagram behaves like a chemical reaction diagram or network. So that kind of diagram is a picture. It also can be thought of as a chemical formula. S becomes I becomes R. And so those arrows are behaving, in this case, as a transformation or mass transport model. And that arrow with a pointed end classically represents transformation or mass transport. In the case of a chemical reaction here, two ADP molecules go to ATP plus AMP. In that case, the interaction is reversible, and so this arrow points both ways. In the case of my simple infectious disease model, S goes to I goes to R. Those are unidirectional. A link is always associated with a rate of action, although we don't always write that rate of reaction on the link. And if we want to take this diagram and make it into something that we can solve either mathematically or computationally, we have to say what those rates are. Okay. So if we have a transformation model, S goes to I goes to R, what we want to know is how they change in time. And to do that, go from a diagram to a mathematical set of equations, the first thing we have to understand is what SI and R mean. And if it's a population model, then SI and R, the names, susceptible, infected, and resistant. And classically, and I'll have to be careful about this because SIR models don't typically use this. Herbert's textbook now is pretty good about this. If you have Roman S, I, and R, those are the names of the species. If you're italic S, I, and R, that's the amounts. So italic S is the number of individuals of type S. Italic I is the number of individuals of type I. Italic R is the number of individuals of type R. And so the first thing we have to do is say we have these names. And now we're going to give numbers, the states of those nodes, which will be populations in this case. And then if we want to actually make this actionable, we have to have a rate of transformation. There's a rate at which S becomes I, becomes R. Those rates have to depend on something. Typically, they're going to depend on the amounts of something, and classically, they'll be the amounts of the things at either end. If it's a chemical reaction and it's a forward reaction, it can only depend on the thing at the source of the arrow, not on the thing at the target of the arrow. In the case of an epidemic model, it can go both ways. The dependence can go both ways, which is why I've written it that way. And classically, you use the letters V and W for rate laws because they stand for velocity. So there's a velocity at which S become I, and that velocity depends on S, the amount of S and the amount of I. There's a velocity at which I becomes R, and that depends on the amount of I in the R. Okay. For a population or even for molecular reaction, molecules are discrete objects, people are discrete objects, and therefore these S and I are in fact integers. There are 55 people in S and 36 people in I. There are 460 times 10 to the 23rd molecules of S and 2.1 times 10 to the 21 molecules of I. So they're integers. In most of the modeling we're going to do, we're going to assume that the numbers of those is large enough we can treat these as continua. So we're going to treat them as real numbers. And that assumption works sometimes and doesn't work at other times. In the case of a virus infection, that infection usually will start out with one individual. And to assume that one individual is a continuum is actually a poor assumption. And so that's something we're going to have to think about. But in general, the models we're going to work on to begin with are going to be models where we assume that populations are continuous rather than discrete. We're going to assume that the rate of change of populations is averaged over enough individuals that it's deterministic. Again, that's not true when numbers are small. And we'll talk about how to do stochastic modeling and see what the differences are. They're quite dramatic. In the case of a disease like COVID, ironically, that stochasticity means that it's less infectious than you might expect. You'll hear a lot about super spreaders of COVID, but in fact, they're not the ones who are the big deal. It's the average population that matters. And then we're going to assume in this case that over the volume that we're looking at, that the connections are uniform, that there's no internal structure. And again, in epidemic models, that's often not the case. People like Alessandro, Vespignani, and Flamini have worked on spatial models of viral infection, and models of things like infection due to airplane connections. We're going to assume that within our little world, the population is well mixed. We will look at some of Perlson's model where there are boxes where you have different well-mixed populations that are connected to each other. That's something we'll get to a little bit later. In chemistry, that's called a stirred reactor model. It tends to use the same name in biology too, although it's not quite as relevant. Okay. And so once we've done this, we're going to write the rate laws, and they're going to be written as simple first-order ordinary differential equations. And they're always going to have the same form. There's only one kind of differential equation you need to know for this class. On the left-hand side of the equation, it's going to be d by dt of an amount. On the right-hand side is going to be a rate law. So if S is going to be turning into I, ds by dt, the rate of change of S in time, is minus the velocity of reaction Vf of S and I has input coming from S, so there's a mass balance term where that minus Vf S of I turns into a plus Vf S of I for di by dt. And then I turns into R, so there's a minus W Vf of I comma R. For R, there's a mass balance term. dr by dt is equal to Vf of I comma R. You'll notice, as I said beginning, there's no entry or leaving of people into this model. So if I add ds by ddt plus di by dt plus dr by dt, it's 0. So the sum of these is 0. If I add a source of new people migrating in, then there'd be an extra term in the ds by dt that wouldn't balance out. If I had people dying and disappearing, turning into nothing or leaving, there'd be lost terms. But in general, in epidemic models, you have mass balance. Chemical reactions, we tend to have mass balance. So we've written a mathematical structure now. We've taken a structure for a network, and we've turned it into a mathematical structure. But until we say what VF and WF are, what the functional forms of those rate laws are, we can't do anything more than that. This is what I was talking about model structure. And so we need to have some hypothesis about the rate laws. And let me come back. Let's see, this is okay. So I had wrapped up a minute ago by talking about the idea of S goes to I goes to R and a rate law. And then I showed a slide that didn't belong there. It's the leftover from an earlier version of the deck. And I want to come back with something which is now going to be even simpler than what I wrote. I'm going to, as I mentioned, the letter S for susceptible turns into the variable T in a lot of these models. So we're going to think about a one-line model of viral infection. And we're going to say that we have a certain number of individuals who are uninfected, but could get infected. Those individuals could represent cells or people in a population. And we're going to call the number of them T1. Or just T, actually. Let's just say T. Sorry, the 1 is wrong. T is the number of uninfected individuals, cells or people. And we're going to say that they become infected. And we're going to call that number I1. And we're calling it I1 because later on there's going to be an I2. And we want to know what the rate of infection is. And that rate of infection is going to be proportional to the amount of virus. And the virus is not going to be a node in this case. It may be later on. At the moment, that amount of virus is just going to be a parameter. We live in a world where there's a certain amount of virus, and that virus is going to be infectious. And the rate at which an individual, whether that's a cell or a person, becomes infected is beta, a rate constant, which is the infectivity of the virus, times the amount of virus. So if I'm an individual, the probability per unit time that I get infected is beta times V. Beta times V is just a number. It's a parameter. Okay. And if I have T individuals, whether that's cells or people, the total number of new infections per unit time is the number of susceptible individuals, T, times beta V. And so now I know my rate law. T goes to I1 at a rate beta times T times V. Notice beta and V are parameters. T is a variable. This actually makes some sense if I think about it. If I had no virus, I shouldn't be getting infected. So this rate is 0 if there's no virus. And if I have no people to infect, if T is 0, then the rate of new infections is 0 too. This happens to have the form of a chemical reaction. It's called mass action law. And it has the nice property that it's linear at 0, and it goes to 0 at 0. It means that my amounts will never go negative. I can't have minus 10 infected people. I can't have minus amount of virus. And so I want a set of equations that don't go negative on me. This has that property. We could actually argue that in the case of cells being infected by virus, the virus is used up during the infectious process. In fact, usually the amount of virus is big enough that that's not significant, but there are times when it is. And in that case, we'd have to write this in the form of a chemical reaction, which would be T plus V goes to I1. And that would have rather different properties. Actually, Josh, we should make a note of that and add that as a homework assignment to compare those results. Okay, so I've now written my rate law. T goes to I1 at a rate beta times T times V. And so that turns into a pair of differential equations, one for T and one for I1. dt by dt equals minus beta T times V. di1 by dt equals beta T times V. Notice that if I add these up, T plus I1 is the number of people I started with. If I started out with T0 individuals and no I1s, then the sum of T and I1 is always T0. So there's mass conservation, people conservation, cell conservation. And that fundamentally is about as complicated as things are going to get, although the functional forms will get a little bit more aligned. And so the idea here is we started with an arrow diagram, T goes to I1. We thought about what's a reasonable rate law to put on that arrow. Once we put that rate law down, we can translate it immediately into simple first order ordinary differential equations. Now, in the case of dt by dt equals minus beta TV, that's the simplest possible differential equation we can write down. If we assume that the initial number of susceptibles is T0 and the initial number of infecteds is 0, we can immediately look at that and write down the solution, which is the T of T, the number of infected susceptibles as a function of time, is our initial number of susceptibles times e to the minus beta v times t. And if people find that level of ordinary differential equation confusing, I encourage you to review the very basics of ODEs. We're not going to assume that you know complicated ODEs, but it's important to know that an exponential ODE looks like that. And if people need references to basic mathematics or refreshers, I'm happy to provide them. So actually, I know the complete solution to this problem already. I don't need to write a computer program to do it. But let's use this as an example to get into computing. And in this case, since we know what the answer is, we can tell whether our computer is giving us the right answer or not. One problem with computing is that people have a tendency to start coding without thinking about what they're doing. And then once you've written a computer program, it gives you an answer. If it crashes, you know there's something wrong. If it doesn't crash, it gives you some answer. If you haven't thought carefully about what it's doing, you may not know whether that answer is reasonable or not. So in this case, it's nice because we know exactly what the answer should be, so we can tell whether what we're getting out of it makes any sense. So now we're going to use Tellurium to solve this problem computationally. As I mentioned, Tellurium is a very simple language for model specification. We're going to run it in class on NanoHub. However, you can run it locally on your desktop machine. I strongly encourage you to use the NanoHub deployment because it'll be consistent. Everyone will see the same thing. And so the first thing we're going to do is, has everyone created a NanoHub account? Has anyone not created a NanoHub account? If you haven't created a NanoHub account, please do it right now because you're going to need it. So if you need me to wait and help you to create it, that's fine. Okay, if you've already created it, I will assume you've created it. Go to NanoHub. You select log in with an affiliated institution, and you should be able to use your IU username and password to get online. Once you're in, and I need people to do this more or less in real time. So then this is something now, what you're going to have to try to do if you're on a computer with a single screen, is what I will ask you to do is do a split screen where the Zoom window is on one side and you have your browser open to NanoHub on the other side. So you can see what I'm doing and NanoHub at the same time. So I'll give everybody a minute to get that working. If you have problems with that, let me know, or Josh know, and he can help you out. But I need everybody to get onto NanoHub now. So I'll wait. I can do a poll if you like, but I need everybody to get onto NanoHub now. And I can't go forward until everybody's on. So if you use the buttons, buttons in the participant list for yes, yeah, if somebody says they're on, I need everybody to say yes before I go on. 2, 3. The people need help. Jeffrey says, okay, you're on. Okay, anyone else? Devin, Antonio, Tyler, Brian. Anybody else? Does somebody need help getting in? Brian, do you need help on getting onto NanoHub? You're on? Okay, good. Anybody else? It's going to be a boring next hour if you're not on, so it's really important to be able to. Is everybody on? Is anybody not on? Maybe that's the best thing. You'll find that once we start doing exercises, I'll ask people, do you need more time? And I'm not trying to hurry you when I ask, do you need more time? But I don't know how long it takes people because there's a lot of variability. So when I ask, do you need more time? It really is a question, do you need more time? Saying yes is not a wrong answer. Okay, so is everybody on? Anybody not? Okay, good. So now once you're in NanoHub, you should see something that looks like this. NanoHub is not the most brilliantly designed website in the universe. It's not the most convenient, but it works more or less. You'll have something called a dashboard, which is essentially a home page. And to get that dashboard, you go to the button where it says login. So first log in. I'll assume people are logged in. Once you're logged in, go to search. If you haven't done this before, it's the first time, go to search. And type Tellurium in the search bar. And so everybody should do that. And you should pull up a screen that looks like this. It says resources. And once you've gotten to resources, there should be a button that says launch tool. Does everybody have that? Because that's the next thing that we have to do. So let me give you, if people can use the yes button to let me know if you've gotten to that. And if anybody has any problems, click no and Josh will help debug. Mercedes is okay. Anybody else? Everybody, anybody? Again, if you don't tell me, I can't determine.
I'm not seeing the screen that you're showing.
So, did you get to this page?
Yep.
And you logged in?
Mm-hmm.
Okay. And so once you're here, click search.
Yep.
Okay, when you get to search, type Tellurium.
Okay.
And it should pull up a tool like this. You see that? Did it find it?
Oh, yeah.
And now click on Tellurium here. You can also go up where it says logged in and we can search it a different way. There are a couple of options. Once you found it once, there's a favoriting button that will keep it pinned to your dashboard.
Okay, I think I'm here now.
And so once you've seen that, you hit launch tool. Once you've launched it once, it should be there available for you. And if people like, I can switch and do it on my computer. And I can walk you through it. But okay, so you're able to get, anybody else having trouble with that? So once you've launched it, let me actually pull up the screen. I'm going to switch to that. Give me 1 second. I have to go back in because since I logged in earlier, it's thrown me out because I wasn't in enough. So let me just show you here. So this is my dashboard. If I go up here to logged in, can you see that? There's a button called dashboard. If I click on dashboard, it'll take me back to this page here. If I want to do a search, I could do the search the way I did. I can also click here on this button, All Tools. And when I go to All Tools, I can do search. I can type Tellurium here. It pulls up Tellurium. I can go click on Tellurium. And then you see this little red heart. It's probably a little small, but there's a little red heart. If I click on that little red heart, it would when you do it, it won't be red. If I click on that red heart, it favorites it, and that'll pin it to my dashboard. So then I don't have to search it in the future. It's there. And I can then go to favorites, and I see Tellurium here as a favorite. And if I click on this box here, launch Tellurium, I'll get Tellurium running. It takes a minute to launch. Once it's launched, it's pretty fast, but the turn on is a little bit slow. And let me make sure that you're seeing what I want you to see here. And what you should see is something that looks like this. Does everybody see something that looks like this? It should say NanoHub Jupyter Tellurium up at the top. There should be two boxes, import Tellurium as TE, and then an empty box. Does everybody see something that looks like that? Yeah, okay. Is anybody having trouble getting to that? Because we're going to be using this a lot. So you'll have plenty of practice doing it. Okay. And in the future, I will probably have the Tellurium window and the slide deck open simultaneously. But again, they get sort of small. The fonts get small. So for today, I thought I would switch back and forth. Once we're doing more programming, I'll have them both on the screen at the same time. Okay. So you've got, everybody has the tool launched. And the first thing I'm going to show you how to do is upload files. Tellurium in principle lets you save things in your local workspace, but you shouldn't count on it completely. And so you want to be able to upload and download things. So what I'd like people to do is, I'll just walk you through how to do an upload. We'll upload this simple model of viral infection that I've got stored. It's in the Google Drive. It's in the shared Google Drive that I've given you, and the link, see if it'll let me copy it. Let me get the link for you. I'll put it in the chat. Hope this works for people. It's also on Canvas. I also have it on Canvas. Did that link work? Once we've done this once or twice, it'll be pretty automatic, but the first few times it's a bit of a pain. Okay, and inside of a NanoHub, you want to click on, there's a missing slide, my mistake. If you go, here it is, okay, fine. So we're going to upload. So the slide, sorry, the slides were out of order here. So we're going to do an upload. To upload, you have that window that says NanoHub Jupyter Tellurium. You're going to go to Open in the File menu, and it'll pop open a new window, and that new window should look like this. It'll say NanoHub Jupyter at the top. It'll give you a bunch of directories, and you click Upload. And we can do this together. I'll walk you through it, and then we'll do it together. You click Upload, and that'll give you a file browser. and you can point it to the shared Google Drive. Or if you've downloaded that file to your desktop, you can point it to wherever you want. It may be easier to download it to your local machine. If people have Google Drive running, then you'll have it available as a local file. If you don't have Google Drive running on your local machine, then it won't. Then you'll have to. Then you probably have to download it. Okay. So I'll give people a second to do that. Once you've done that, it'll show you the name of the file you're trying to upload. It doesn't actually upload it yet. You have to hit the upload button to do the upload. And this slight inconvenience is there basically because you're using somebody else's server and they're relatively finicky about what they allow to be uploaded and downloaded to their server because you're running on their computer. And so that means that the process of uploading and downloading things isn't as straightforward as it might be. Were people able to do that, to upload? All right. Is anybody not able to get it? Everybody seems to be okay. All right. Okay. Once you have it there, it should say Simple Model of Viral Infection, Lecture 1, Exercise 1, IPYNB, means it's a Jupyter Notebook. Double click on that, and it should pop up a little window that looks like this. Did that work for people? Does anybody need help getting that running? If you do, we can always do a breakout room, or we can just go over it together. Okay. I don't know how much people have used Jupyter Notebooks. You can either hit Shift and Control, or Shift or Control Enter, and that runs cells one at a time. Or you can pull down the cell menu and type Run All, which will run them all sequentially. Okay. If you run this, it'll display the results of the simulation, which is that the number of susceptibles goes down exponentially in time. The number of infecteds goes up exponentially in time. The sum of these two is constant. Does everybody have that? All right. If you want to download things, the download is actually more convenient than the upload. You should always download any programs you've written on NanoHub. They should be persistent, but no promises. Again, you're on somebody else's computer. You don't have control over. If you go to File, hit Rename, give the file a name, whatever it is you like, then hit Save and Checkpoint to store it locally. And then go back to File, select Download As, Notebook, and then that'll download it to your Downloads folder. And you can move it anywhere you want on your local machine. So you don't have to go through that second window, which you do on the upload. You don't have to go through that open dialog. Okay. If for some reason you want to run this in a different Python IDE, like Spider or raw Python or something else, you can download it as python.py. That'll give you a plain text file. The Jupyter Notebook file has formatting information in it, so it's sort of ugly to look at as text. I don't know, how many people have used Jupyter Notebooks before? 1, 2, 2, 3. Okay, so many people have. We're not going to be doing anything fancy with it. It's relatively easy to get used to, I think. I happen to actually like Spider better than Jupyter Notebooks, but what we have online is Jupyter Notebooks. It works fine. Okay, any questions about this? We'll do this a lot and we'll go over it again, but essentially this is the level of detail. The one thing we may run into a little later on is that when you upload and download files, well, the upload, the open command, puts the files in your local working directory. And you may want to move files around. It's a Linux box, so moving files uses commands like cd, ls, mv, so Linux commands. And we'll come back to that later. At the moment, we won't have to deal with it. Okay, so we're now inside of Jupyter Notebooks, and you should have a screen that looks like this with some stuff in it. So the native language here is Python. Boxes are interactive cells. There also are markdown cells. You'll see that first in the let me actually go to the window which I have this loaded up in. In the image that you have, you'll notice there's some formatted text at the top. It says we start with a population of uninfected susceptible cells. That's a markdown cell for text. It actually has tech in it. And then we have a cell which says import Tellurium as TE, and then some other stuff. And then below that should be a display. Okay. And I remind you that to run a cell, to run an individual cell, you hit Shift Enter when you're inside the cell. If you want to run all of them, you go to the cell menu and hit Run All. One thing that we'll run into, and it's not going to be that important now because we're not going to be saving a lot of things, is that the first time you open Jupyter Notebooks, it may say at the top, read only. And if it says read only, that means that you can't actually save your file. And so if you're getting a read only message at the top like that, the first thing that you can do is you go to File, New Notebook, select Python 3, and then that will then open a new window, which is no longer read only. And the only annoyance there is that you have to retype import Tellurium as TE. And very often we'll use NumPy or library. So I usually will do import NumPy as MP as well. Are people seeing that read-only message on their screens or not? It is. So if it is, then go to File. Well, actually, for today, it should be all right. I don't think you'll have a problem with it. But in the future, when you start Tellurium, if you get that read-only message before you do anything else, restart the notebook. I think you'll be okay today. Okay. So now that you've got that uploaded, does everybody see something that looks like this? So you've got the exercise. It doesn't have it doesn't say exercise 0, but it says okay. It has slightly different text. The one I have has slightly different text from this, but it has a set of code in Python. And if you run it, when you run it, does it give you a graph? Does it plot? Does anybody not have it working? If it doesn't work, speak up, please. I'll assume it works if everybody doesn't. Nobody says anything. I'd like to walk you through what this code is, because this is about the level of complexity that we're going to have for the programming side of things for the whole class. The thing that we're going to wind up doing is doing some loops and data analysis in Python. But as far as writing models goes, this is about how complicated it gets, which is not very bad. So if you want to learn about Tellurium, we've seen this before. There's tellurium.readthedocs.io, which has this very nice online tutorial. And I strongly encourage you to cut and paste some of the examples from the tutorial into your NanoHub Tellurium and run them. So let's walk through this program. This is implementing this incredibly simple network model, which has 2 nodes and 1 arrow in it, and a rate law that is got 1 variable and 2 parameters. We see the line here we see the line here, import Tellurium as TE. Tellurium is a library of Python functions. And TE is simply a short name for it because typing Tellurium all the time is inconvenient. So import Tellurium as TE imports the Tellurium library, which we're going to use a lot. Our model definition is in the form of a character string. That is quite flexible. It has some inconveniences because the parsing is not going to always be informative if they're typos. So the actual model definition is in the form of a string. And in Python, if you have strings which have carriage returns in them, you need to encase them in triple quotation marks, either single or double quotation marks. So we're going to say model string, which is going to be a string that is going to contain our model definition, equals, and then a triple open quotation mark that's going to be balanced by triple closing quotation mark at the bottom. Our model is defined between those opening and closing quotation marks. And then we're going to have a little bit of simple Python that's going to load that model, this line, run that model, and display it. Okay. That means that every simulation we do is actually going to have two languages in it. It's going to have this model specification, which is written in the antimony language. And it's going to have the thing that actually loads and executes that model, which is written in Python. And occasionally we'll have things that are inconsistent, slightly inconsistent between Python and antimony. For example, exponentiation in one case is double star, and in one case is a caret. The comment command in Python is a hash symbol. In antimony, it's a double slash. So fortunately, there aren't that many of these inconsistencies that we have to get used to, but there are a couple. Okay, so let's walk through this little program. The first line is import Tellurium as TE, which we've already gone over, and that loads the library that we're going to run. The second line, model string equals double quote, close double quote, triple quote, blocks out the string that we're going to put our model in. And the yellow is the actual model specification. What we're trying to write is this very simple model. T goes to I1 at a rate beta times T times V. Antimony was designed to write chemical reactions and mass transformation. If we write this as an ordinary differential equation, we have two of them, one for T and one for I1. The fundamental concept in antimony is a transformation law with an arrow. It's trying to look like an arrow diagram. And so we're going to write that there's a reaction or an equation E1, which is T turns into I1. The name of the equation is followed by a colon. The specification of the equation itself is written with arrow. And then there's a semicolon. The semicolon may be not obvious. And then the rate law, which sits on top of the arrow, beta times T times V, and then a semicolon at the end of the line. If that looks a little bit like old-fashioned C or C++, that's where that semicolon notation. Double slash is a comment. So we're going to say our equation, we only have one of them. E1 is the name of the equation. We don't need the name, but it's useful to have it. T goes to I1, semicolon rate beta T times V. And that actually gives us two ODEs, the decreasing ODE for T and the increasing ODE for I1. Okay. That's
so,
all.
Professor,
Yes, please.
if we were to have three nodes like the SIR, would that be in two separate lines as two separate equations, or would you just list this? Okay.
So each arrow gets its own line. So if I had a source term where something came in and made T, I'd have two lines which had T in them. If I have I1 created here and disappearing there, I would have E2, I1 goes to I2, and then the rate. Now, we're not going to be, in this particular case, we're not going to be using what's called stoichiometry. But if I had a complicated chemical reaction like 10 O2 plus 6 ATP plus 3 enzyme goes to 16 molecules of species X, those numbers are kept track of, and it will automatically build the ODEs for you. You don't have to do the mass balance by hand. And so if you're doing complicated chemical reactions, Tellurium writing it this way is very convenient. If you're used to writing ODEs the way you would in MATLAB, it takes a little bit of getting used to. You can, in fact, write everything as an ODE. If I wrote nothing goes to T at rate minus beta TV, and nothing goes to I1 at rate beta TV, that would exactly replicate my ODEs. So I can write ODEs with this, but it's designed to do mass balance and mass transformation. And it may take a little bit of getting used to for that. But in the end, it winds up being pretty convenient to do it this way. Okay. And yes, if I wanted to write it as ODEs, I could do it this way. E1 goes to A, T goes to nothing at rate beta TV. E1B, nothing goes to I1 at rate beta TV. That would be another way to write it. So I have some flexibility how I do it. Okay. So just in the analysis, which I've already gone through, E1 simply gives a name to this rate law. T goes to I1 specifies the chemical reaction or the transformation. Beta times T times V specifies the transition rate. And if I had T goes to I1 goes to I2, I'd have another line. And in principle, if I had hundreds of arrows, I could have hundreds of lines in my code. The one thing that you can't do is dynamics of networks. You can do it, but it's pretty awkward. So if I want my network architecture to change, this is not the best way to do it. In other words, this is really designed to specify a particular network and then have dynamics on the network. If I want to add or subtract links in the network, it's a little bit cumbersome. But we're not going to run into that particularly in this course. There are a variety of technical reasons why this language has the form it does. If you write the ordinary differential, so if I write the equations in the form of arrow diagrams like this, I can always derive the ordinary differential equations unambiguously. If I write the ordinary differential equations, in this case, if I write dt by dt equals minus beta TV, di1 by dt equals beta TV, I can unambiguously rederive this arrow diagram. In the case of complicated arrow diagrams, you cannot go back from the ordinary differential equations to the underlying arrows. And this is a homework assignment I would give if we were doing more of a network, of a biochemical network course, which is there's a beautiful paper by John Tyson on cell cycle. And John is one of the great mathematical biologists, and he has a stack of ordinary differential equations that describe how cyclins work inside of a cell. And the assignment for the students is to read his paper and tell me what the chemical reactions are. And you cannot do it. You cannot determine what the chemical reactions he represented were, because the ODEs do not allow you to back out the chemical reactions. And so Herbert, who invented this language, is a strong believer that things should be reversible and that the information of the model should be there in the code. And so he created a modeling language that allows you to maintain the network architecture. And that's what's sort of special about Tellurium. Other languages don't have that. Okay. Now we have to specify all the parameter values. There's only one parameter here, beta, the infectivity. And we could explain why is it 6.2 times 10 to the minus 5? Well, this is actually derived from a model for flu where that was measured. So it's actually experimentally valid. And it's always a good idea when you write a rate law to actually say what the units are. When one's playing, one tends to use a number like 1 and not think about what the rates are. In this particular model, it comes from a flu model. Time is measured in units of days. Amount of virus, which we don't really have yet, is measured in a unit called TCID, which is the amount of virus to get half of the cells infected. And so the rate is 6.2 times 10 to the minus 5th rate of each of one cell becoming infected per day per unit of virus, TCID unit of virus. And I have to specify initial amount of virus. In this case, it doesn't change. It's a parameter. And it's 10,000 units of TCID. Doesn't really matter what that is. Because what matters is the product. So 10,000, 10 to the 4th times 6.2, 10 to the minus 5 is 6.2, 10 to the minus 1. So basically a cell has a probability of 0.6 per day of becoming infected. I have to say how many cells I initially have infected, 10 to the 7th. Again, for our purposes, it could be anything at the moment. Again, comments are double slash. Initial conditions, T0 initial number of cells. I'm writing T0 equals 1e 10 e to the 7th, T equals T0 rather than just T equals 1e to the 7th, because I may want to refer to the initial value later on. The variable T will get overwritten, and so I'm saving it into a parameter. And that's the basic syntax of antimony. Now, antimony has all sorts of other things that you can do. You can have events where things happen in time or under different conditions. You can define functions. You can define all sorts of things. And Herbert's book has, and the examples online have all sorts of nice teaching of that. And I'll teach those as we go along. There are a lot of functionality in antimony we won't use. It's quite elaborate language. But the one thing that you'll find with antimony, it takes sometimes a little bit of thinking about, is has anybody used a declarative language before? So this is a sort of a philosophical question. Most languages are procedural languages. Python is a procedural language. MATLAB is a procedural language. It specifies things to do. Antimony is a declarative language in the sense that it specifies a model structure. So we're specifying a network and how things interact on it, but we are not actually specifying a dynamics. There is no sequence through which lines are executed. So if I type an equation E1 here, and then E2 and E3 and E4, those aren't conceptually executed in sequence. They're executed at the same time. And this language is not specifying actually the program's execution. It's specifying a structure of a model. And that may not be very important right at the moment, but it means you can't do certain kinds of things. There's no concept of a loop in antimony because there is no sequence of execution in antimony specifying a structure. And we'll come back to that later. So if you want loops, you have to do them in Python. All right. Now, once we've specified our model, we have to go into Python to do something with it. And the standard thing that we're going to do is convert that antimony string into something that's executable. And so te.load A is something we're going to get very familiar with. TE, Tellurium. Load A stands for load antimony string. So te.load A, and then the name of the string, loads and parses the model that we've written in antimony. And we're going to make that available with a handle M. So M is now a handle to an executable model, an executable Python executable version of the model that we've specified. All of the attributes of our model are available using standard Python syntax in the object M. So if I want to know how much virus I have, if I've loaded my model in m.v returns the amount of virus. m.t0 returns t0. m.t returns the amount of susceptible cells. m.I1 would return the number of infected cells. So it's a very simple way of referencing between the antimony model and the Python. Does that make some sense? We'll play with that, so I hope everybody will get used to it. If people have used Python before, in Python objects, dots should be at least vaguely familiar. If not, we're not going to use anything more complicated than this one level of dotting, so it should be all right. The next thing we have to do is run the model. Remember that antimony specifies the model. We've loaded the model, we haven't executed it. And Tellurium has a couple of methods for simulation. The main one is m.simulate. So simulate is a function, it's a method actually, that operates on M, the object, the loaded model, and runs it. And it returns an array, which is the output of that simulation. So S equals m.simulate executes the model we've loaded. And it has 3 arguments. It potentially has a lot of arguments, but we're always going to use 3. The starting time, here is 0. The ending time, we're measuring time in days, 10 days. And the number of points we're going to use in our simulation. So we're using it, this happens to use an explicit time step with an explicit time step. And one thing we will have to change, depending on what we're simulating, is how many substeps we're going to use. If we make that number too small, the thing will crash. Okay. And so now S is an object that contains the output of our simulation. And we could print S. We could, if you go into your code here, why don't you try this? After S, well, you could just go into a cell and type S and return. Were people able to do that? I'm going to do it on my screen. Let's see if it works. If it doesn't work, you can type print S. Yeah, S works. So if you go into a cell below the current cell and type S and hit return like this, what you'll get is something that looks like this. And I'd like people to try doing that. Everybody okay with that? And what you'll see is that S is an object that's got a somewhat complicated structure. It's got square brackets, so it looks like a list. And it's a hybrid list because you'll see at the top, it has columns that are labeled. So the variable names, time is a reserved word for the time. Time, the variable t, and the variable I1 are labeled. And then the values are in these columns. That's great because the output contains the information about the names of the variables. It's convenient. It's a little bit less great because it's not a NumPy array and it's hybrid. So if I try to then do arithmetic on this thing, it'll crash because it's got these strings sitting at the top. And so if I want to use this as an object to do calculations on, I have to strip out that first line. Today, we're not going to do that, but at some point, we're going to have to learn that. Okay. And then typically, we'll use matplotlib plotting functions, but there's some basic plotting functions that are built in that know how to handle this object. m.plot use a Tellurium function that will plot this object that we've got. And it'll automatically use the information to label the variable names, T and I1. And there are a variety of options for that plot. You can use log scale, floating axes, fixed axes, name the axes, and so on. And they're all in the documentation, online documentation. But for the moment, we'll use the default plot, which does what we want. Okay, so now, boy, we may end class a little bit early today. We can have some discussion and ideas. Why don't we try using this simulation? So why don't you try changing the value of beta and run it? We start out with beta 6.2 e to the minus 5. Why don't you make it 6.2 e to the minus 4 and run it again? See what happens. And I'll point out you can do that in two ways. You can either go in and change this value here where beta is specified, or after I do m equals TE load A, I could write m dot beta equals and the new value. That will overwrite it. And the fact that you can do that overwriting is part of the introduction to why declarative languages are different from procedural languages. In general, in a procedural language, I can't overwrite part of my my program specification that way. Here I can. And I guess I can switch to screen sharing my code, the code here. So I'm going to check, I can change beta here. Can people see my programming screen now? And I can run that. If I make beta bigger, of course, the cells become infected faster. Let me show you how to do that here. M dot beta equals, let me make it some different number, make it 10,000. That's huge because 10 to the minus 6 is very small. So it happens very fast, 0.001. So I can change beta either in the model specification or outside of it. So why don't people try playing with that a little bit until they're comfortable with it? There isn't that much this model will do. Basically, you can change T0 and beta. So why don't we take a few minutes? Okay, yeah, it becomes wider. If you make beta smaller, it'll get wider. If you make beta larger, it'll get steeper. If you change T0, it'll change the axis label, but because it's auto-scaled, it won't look like it does very much. So if you make T0 1 e to the 8th, e to the 8th instead of 1 e to the 7th, and you redisplay it, the only thing that will change is the label 1 e to the 8th at the top. It'll look almost identical. Why don't you try that? The other thing that's sort of interesting is we're using 100 intermediate time points here. If we tried to do the calculation with 10 intermediate time points, what happens? So that would be coming here to this line m dot simulate 0, 10, 10. Try that. What happens when you do that? Yep, exactly. You get stair stepping. Basically, you don't have enough intermediate points to be able to get a meaningful result. If things were more elaborate than this, if you make the number of points too small, you can get a convergence error where the integrators fail. If you make that number very large, the simulation will take longer to run, but it won't make any other difference. So if you made the number of intermediate points 10,000, It would take a little bit longer to do the plot, but otherwise you won't get anything different. If you make that number really huge, like 10 million, the program may crash because it'll run out of memory. Yeah. Okay. Does that work for people? Do people want to play around with that some more, or should I go on? It's not the most, this is sort of the hello world of antimony programming. It's not the most exciting thing to do. You can't do that much with it, but actually it has a lot in it. Really, in terms of the complexity of model specification, we've got almost everything we need. The only thing we really don't have are events. And they're pretty easy to write. As far as the Python goes, we'll have to do some loops. We'll have to do a little bit of slicing of our arrays. So the next thing I want to do, because we're going to run out of time in a few minutes, is a little parameter scan. Because a lot of what we're going to do in class will be to say, okay, if my level of virus is 10, 20, 30, 40, 50, 60, what happens? And so let's do a very simple parameter scan. So we're going to build a loop that's going to scan the value of the amount of virus in my system. And I'm going to scan it over powers of 10. So I'm going to make it 1, 10, 100, 1,000, 10,000. And one of the things that I will never forgive Python for is that it doesn't have an intrinsic real numbered loop. The only intrinsic loop structure it has is on integers. And so I typically don't use, I use the NumPy arange function instead of the Python range function. But for now, we'll stick with, we won't use NumPy. So the basic loop architecture for Python is for p in list, and range is a list, p in range 0, 6, colon, the syntax. And remember that Python is 0 based, and so this will give us 0, 1, 2, 3, 4, 5. It will not give us 6. So that's something you have to remember. Not like Fortran where it executes the last value. And then we have a specific Tellurium command, reset all. By default, if I call simulate twice, and maybe this is something we should have had as an exercise. If you call simulate twice, the simulation will continue from where you ended. And so if I want to reset my variables and parameters to their initial values, I use m.resetAll. That effectively reloads the model that I've written. Otherwise, it would continue from the last time when I loop. Another way that it's different from a procedural language. I set m.v, my virus amount, 10 to the double star p. I run my simulation. And then another one of these things that's a Python-y thing, m.plot, show equals false. And that will cause the plots to be overlaid on each other. Show equals true would give me a separate plot for each one. You can try that. And then te.show displays the things I've accumulated. There are a lot of other ways to do stacked plots, but this is the simplest one. Okay, so walking through this again, I create a loop for p in range 0, 6, colon. I reset my parameter values in my model, m.resetAll, m.v equals 10 to the p. I set my virus amount with that. I run my simulation. I add the plot to the stacked plot without displaying it, show equals false, and then I display it. Okay. if I'm going to overwrite, if I'm going to do a lot of stacked plots, I may not want to plot every variable together. And so one other thing that I might want to know is that Tellurium allows you to select which columns you want to display, which variables you want to display. And the way you do that is at the end of the simulate line, I put a comma, and then I have a list of the things I want displayed. So it's a square bracket, time, which is a reserved word, comma, the variables. In this case, I'm getting everything. Time, t, and I1 is all I've got. But if I wanted something simple, I could plot time and just t. If I didn't put time in, it wouldn't do time. I would have no time column. And then when I plotted it, I would get time t versus I1, which you could try. So why don't people try that? Why don't people actually type this code in and see what you get? Is that big enough that people can see it? I'll do it in my screen at the same time. But this will be your first little programming exercise.
So when I don't include time, it just kind of throws an error.
Okay, probably because m.plot doesn't know what to do if it doesn't have the time column.
Okay, that's good.
Well, did you put t and I1 or just t?
T and I.
Okay. I have to say I hadn't tried that, so I will double check that. M.plot probably doesn't know what to do if it doesn't have the time series. This is what I get when I run it. Did somebody get something that looked more or less like that? Anybody having trouble getting that to work? And let me try what happens if I try to select the columns. Yeah, didn't like it, did it? What was the error? Cannot plot time course. First column is not time. Well, at least it's an informative error message. So if I wanted to do that, I'd have to use matplotlib. All right. Okay. And one could cut and paste this twice and plot all the T's on one axis and all the I1s on another axis like that. And I'll let people play with that a little bit. So what you could do now is we change the amount of virus. If you want to change beta, That's pretty easy. All you have to do to change beta instead of the virus would be in this line here. Instead of typing m.v, type m.beta. Or if I wanted to change the initial amount of t, I'd type m.t0. Why don't you try doing that very quickly? It doesn't take, it just takes a few seconds to do it. I'll do it in parallel. Changing beta gives me, those values don't make a lot of sense. I'd have to make it beta to the minus 10 to the minus p. If you change beta, beta is a small number. So you probably would make m dot beta equals 10 to the minus p there. Would make more sense. For t0, see what happens if I make it t0. If I do t0, only the biggest one makes much difference. Were people able to get something useful out of that? Did it run? Again, it's not very informative because the model is too simple, but it should work. Okay. Okay. So that's all as far as I wanted to go today. I realized we didn't do much in the way of either mathematics or biology or coding, but I hope one or two people had said in their questionnaires that they were worried that they might not have remember Python very well, things like that. I hope that suggests that the amount of Python we're going to use, it'll get to be a little bit more than this, because slicing, you have to remember iterators and things like that. It's not going to be a lot worse than this. And the model specification is not going to be a lot worse than this. But what will change is that the models will get a lot more complicated. And we'll have to think pretty hard about what we're doing. And when we do optimization, we'll use some relatively fancy NumPy optimizers. And those have to be wrapped into functions, so that can take a little bit of effort. But fundamentally, most of the things we're going to be doing are not going to be mechanically much more complicated than what we've done here. Intellectually, thinking about the meaning of this can be pretty significant, but mechanically, it's not going to be that hard. So I hope that gives you a bit of a background into this. Now, let me just show you what I have on the homework assignment. And I want to say a couple of things about homework. The first thing I want to say about homework is that the homework assignments are written before class. And I don't always know how far we're going to get. And so if there is a homework problem that is about something we didn't cover in class, you have two choices. You can either try it, in which case you'll be very well prepared for the next class, or you can say, okay, we didn't cover this. I'll defer this problem and assume it'll be reassigned next week. So if there's a problem on the homework that we didn't cover, don't panic about it. The second thing is that at the top of each homework assignment, I will indicate approximately how long I expect you to spend on it. And the purposes of homework is to learn. It's not to torture you. And so if I say it should take you an hour and you've spent 7 hours on it and you're stuck, stop. Don't kill yourself. Contact me, write, hey, this is taking a lot longer than I expect. And I ask you to tell me when you hand in the homework at the top, give me a fair estimate. Don't inflate it. Don't say it took you 10 hours if it really took you an hour. But if it really took you 10 hours, let me know, because then I will recalibrate. And don't spend a huge amount of time. I do expect people to work week by week on this systematically, because if you get behind, it's hard. But I don't want people spending their whole lives on this course. You have other things to do. And so I'm going to try to give you assignments that are reasonable in terms of the amount of time they take. Not negligible, but reasonable. And if I accidentally give you a problem that takes forever, tell me, don't kill yourself. The second thing is that problems are going to tend to be somewhat open-ended. And that means that they can be more elaborate or less elaborate. And so people who have more experience or find something easier, I expect them to spend more time and do more of the problem. And if it's hard and you're frustrated, do less of the problem. And yes, you're going to be graded on it. But in general, if everybody does the work for the course, they get a good grade. So don't worry if you got a 10, 15 out of 20 on a homework problem. That's all right. I would ask you to try to get a hold of the textbooks. Everybody seems to have some basic programming, so Roslyn may not be necessary, although it's fun. I'd suggest looking at the beginning of Herbert's textbook. Yi has written this nice document called Paper Review, which I've uploaded. It's in the form not of a document, but of a slide deck. And it talks for people. I don't know how many people have actually read scientific papers. For undergraduates, they may or may not have done that. But she wrote a very nice, Yi is a former student of mine who now teaches in Atlanta. She's a senior faculty member in the math department in Atlanta. And she teaches math bio every year. And she wrote a great paper, a great exercise called How to Read a Modeling Paper. And it's really helpful in giving you some orientation about how to look at a scientific paper. Josh points out in the chat that if you need help, please come to us. So what I'd like you to do there's a list of papers we've uploaded on COVID. And I'd like you to look at one of those, look at Yi's guidance about how to analyze it, and go through that. And I don't expect you to read the paper in detail. I don't expect you to spend 20 hours reading the paper and analyzing it. But I do expect you to look at it enough, spend an hour looking at the paper, an hour and a half looking at the paper, and write up a little summary following Yi's guidance about how to analyze it. And if you want, I could open her slide deck. If we had a little more time, I would have gone through that. The other thing I'd like you to do is go to Google or Google Scholar and search on COVID mathematical computational modeling. And as I mentioned, there are many different places you can use mathematical computational models: epidemiology, viral replication, drug delivery, tissue damage, and so on. Try to find one paper that's not in the list that we have now on modeling COVID. Could be on flu or some other kind of virus too, if you prefer. And I don't expect you to read it in detail, but I do want you to read at least the abstract and look at the figures, enough to say it interests you. And enough to say, just in a few words in your homework, give the name of the paper, a few sentences about what it's about, and why you think it's interesting. And again, I don't expect people to spend a lot of time. I expect you to spend an hour on doing that. And then the third one is to do exactly what somebody talked about earlier, which is we did the first part of an SIR model, which was T goes to I1. But I'd like you to try it. If you get stuck, try it for an hour and a half. If you get stuck, it's okay, we'll do it next week. Is rewrite this to be T goes to I1 with a rate law now, beta times I1 times T, not beta times V times T. And then I1 goes to R at a rate K times I1. And I want you to run that and change the values of beta and K and see what happens. And if you're interested, if you get that Britton textbook on SIR models, he's got a full analysis of that problem. And it winds up that we could fit the epidemiology data, that curve that I showed of the infection rate in Italy or England, not the US, with that SIR model. And we'll try doing that next week. We'll run some fitting code. We won't go into the details of how fitting works, which is a little more complicated. We'll come back to it, but we'll have code. Josh has written some nice fitting code. We'll run that next week. And that'll give us an introduction to the idea of how to connect a model to data. Okay, are there any questions? People have been pretty quiet. You could always, you're muted, but you could always unmute and ask questions. So are there any questions before we break for today? Was that a reasonable start for things, I hope? I want it to be challenging and interesting, but not intimidating. And since I didn't get since I didn't get the questionnaires from some people, I don't know exactly how to calibrate. People are free to send me messages. Gee, you're going too slowly. Gee, you're going too fast. Gee, we need to spend more time on this or that. A lot of the material for this course is stuff we've taught a lot before, but we haven't used this COVID example before. And so there's going to be a fair amount of new material, which means there are going to be some mistakes. And so if you catch mistakes in the slide decks or missing pieces of information, please let me know. And I will update the slide decks. The lectures will be recorded and put online. And the slide decks will be updated if there are mistakes and corrected and uploaded, okay? Anything else we want to go over now before we break? Everybody's been very patient. I know it's a long class, and 5 to 8 p.m. people probably would like to eat. People hungry? I'm hungry. So I hope I'll see you all next week. Again, I can't have physical office hours, but if people want help, Josh is super helpful. He knows this stuff inside and out. In fact, he probably knows it better than I do in many ways. So between the two of us, we'll do our best to make this a fun semester. And again, I know some people may have interests other than COVID, but I'll say for the last time today that the methods you're going to learn here will be very general. And so if you're interested in neuronal modeling or something else, this is a simple way to get into the concepts that you'll need for that. And so you'll find that a lot of the things that we're going to do are not as different as you might think, even though we're talking about viruses rather than neurons. Okay. Anything before we break? No? Well, thank you very much, everyone. I look forward to seeing you next week. If there are any issues with the homeworks, anything else, please let me know, and we'll do our best to make things as easy as possible. And I appreciate you coming and working with us. Last semester, which was the second part of this course, we actually did, 2 of the students started as a project that we started in the class, wound up in a paper. So actually we've got a journal article out of the work that was done. So that does happen from time to time. Certainly not a requirement of the class, but sometimes these models get to be quite interesting and people run with them. So that's a possibility. So I look forward to seeing you next week, and stay healthy. We may be modeling COVID. Please don't experience it firsthand. Has anybody gotten it? No, everybody okay? Okay. One person in my group had it. Vedang, one of the students got it. And he was really sick. It was not pleasant. And one of our regular collaborators, Bob Bacallao, ironically, he's a surgeon up in the School of Medicine. And he got it really early and was really sick. So I know some people are fine with it, but try not to get it, please. So let's study it, but not study it in our own bodies if we can avoid it. Stay safe. Look forward to talking to you next week. Thanks for joining us. Bye.
Thanks, Professor.
Thank you.
Okay, bye. Bye, everyone. Josh, how did that go? Was that all right? I can't hear you.